Yeah, cool. Okay, thanks everyone. So Anatoxin, if this is something that you haven't heard of before, is becoming a larger and larger concern in freshwater ecosystems, really quite a bit in streams and rivers. It's a potent neurotoxin, and it's one of a major class of freshwater cyanotoxins. It's actually been called the very fast death factor. And it's coming up more and more in news articles over the last few years or last decade, where dogs are often, not often, but dogs have been found dead after going to rivers. And it's been attributed to consumption of cyanobacteria that's producing this nerve toxin. And so there's this pretty urgent need to better understand the distribution and ecology of the toxin production from the cyanobacteria. But up to date, most of the research and attention on cyanobacteria has been on these planktonic blooms, like the one that has occurred in the Klamath reservoirs that are no longer here. And we don't have nearly as much information about these benthic or attached cyanobacteria that can produce toxins. And this graph is just showing the number of publications focused on those planktonic versus benthic cyanobacteria. And you can see there's just way less emphasis over the last 30 years on understanding these attached algae compared to the planktonic ones. And so until recently, the detections of benthic anatoxin have been really sparse globally. A 2020 review paper found less than 10 countries had reported anatoxin from benthic or attached algae. But some studies that have these more intensive surveys have found really widespread anatoxin from benthic algae. So New Zealand is where this is most well studied, and a survey of over 100 rivers throughout the North and South Island of New Zealand found cyanobacteria proliferations, which are shown in these pictures here on the bottom, across both islands all over the country. And they found that 75% of these rivers had anatoxin in them. So so even though it's not being reported widely on some scales, on other scales, we are seeing quite a bit of it. Closer to home in the Eel River, there's also been widespread detections of benthic anatoxin from these cyanobacteria. 60% of the cyanobacterial dominated mats in the Eel River watershed, and you can see from this map, this was a really broad study from throughout large portions of the watershed, had anatoxin in it. And this is a river also that's had reported dog deaths. So we have these like two kind of different pieces of information, one saying that, you know, it's pretty rare to have benthic anatoxin, and then these more in-depth studies are showing quite widespread benthic anatoxin. So it really leaves us wondering to the extent that, are they rare, or are we just not looking for them? And so this question came up in the Klamath River. There's been a lot of emphasis on understanding these planktonic blooms from the reservoirs over the last couple of decades. And more recently, there's been some work leading us to start wanting to understand better to what extent these benthic algae are producing anatoxins. So the consortium, the Klamath Water Quality Consortium, that 5-tribe collaboration, they supported a project, 5 or 6 years ago now maybe, where we collected all the anatoxin samples from throughout the watershed that had been surveyed mostly by the tribes over the last couple of decades. And we found about a 6% detection from over 200 samples of anatoxin. So that's pretty low. But all those samples had been grab samples of water. So kind of doing a sample that might not be targeting those benthic mats. In 2016, Tim Otten, who's now with Ben Genetics, did a study on the Klamath River, and they looked for the anatoxin production gene in 126 water samples from 2016, and they found a much higher rate of that gene that's necessary for toxin production, a 30% detection rate. So they also in that study started to try to understand who the toxin producer was, and that information led toward ideas that benthic cyanobacteria might be the ones rather than planktonic ones producing the anatoxin. And so we started just noticing in the field during other surveys these benthic cyanobacterial mats in the Klamath River. And that led to a study that we did in 2021 that was led by the Karuk tribe to understand just some really general general things about benthic cyanobacteria, how anatoxins were distributed through different sites throughout the basin throughout the summer, and also comparing those water column grab samples to actually sampling that material. We wanted to try to understand what taxa was producing the anatoxins and to start to think about what conditions in the river might be promoting anatoxin. In that study, and I'm not going to get into all the details of that study because that I want to make sure I have time for the Quartz Valley data, but in that study, we did find really widespread cyanobacteria from benthic sources in the Klamath River and some of the major tributaries that we sampled. So on the left there, those are kind of macroscopic photos, things that we see in the field. And on the right, you have some microscopic photos of the taxa that we were encountering in the Klamath River. And then these 3 maps, the open circles show places we didn't detect any cyanobacteria, but all the filled in circles are detections of cyanobacteria. So in August and September, we found, sorry, not just cyanobacteria, but anatoxin. So in August and September in this study, we found anatoxin pretty much everywhere we looked, with the exception of just a few sites throughout the Klamath River watershed. And some of the highest rates of anatoxin detection were in these major tributaries, so the Scott and the Salmon and the Trinity rivers. And that was not really what we were expecting to find. And so from that study, we came out with some findings that we were excited to kind of think about in terms of how they can inform monitoring moving forward. And some of the things that we walked away with is that Microcystin was the most likely anatoxin producer. And so visual surveys for anatoxin production from benthic mats should focus on this genus called Microcystin. qPCR was a really effective way to monitor in a lab environment, and it's a cheaper way to process samples than doing direct toxin measurements. We used drift nets, and we found those to be really useful to capture floating mat material and to try to detect anatoxins that way. We also found those anatoxins in those higher water quality tributaries, and so making sure that we're not ignoring these streams that we think of as traditionally higher water quality is important when studying anatoxin from benthic sources specifically. And then we also found that late summer was the time of highest toxin production, so really focusing efforts in that time frame would be a good use of resources. This study and the details of that is going to be published in a September issue of Freshwater Science. I'm happy to talk more about that, or feel free to keep your eyes out for that. But I want to jump into the data collected over the last couple of years by the Quartz Valley Indian Reservation, which was really spurred by the fact that we found some of the highest detections of anatoxin in the Scott River in that 2021 study. So this project was initiated by Crystal Robinson. Bonnie Bennett led a lot of the field work. Sarah Schaefer has been leading the project last year and continuing to lead the project. And then Tuck Super and others at Quartz Valley Indian Reservation also collected a lot of this data. So we wanted to bring similar questions to the Scott River watershed and think about how extensive these cyanobacteria and anatoxins were there. So we asked how anatoxin, again, varied seasonally in the Scott River watershed, what the spatial extent was beyond just those couple of sites in the Scott River. And we wanted to know how we could, again, efficiently and effectively monitor for it, specifically in these Scott Valley streams. Okay, so I'm going to be showing some graphs from the Klamath study and some from the Scott study because they're pretty complementary. And I'll just try to make sure that I'm pointing out which is which here. But one take home from both of these studies is that Microcystin, which is the taxa, it's a genus of cyanobacteria, were the dominant benthic cyanobacteria that we found in the Scott River. You can see that in this green color on these bars here. These are 2 different sample types. They were the dominant cyanobacteria in almost every sample that we found. In the Klamath, Microcystin is actually shown in yellow. These plots are taken from 2 different reports, but they were also quite common, not necessarily always the most common, but quite common. So Microcystin, we also found through some genetic work that Tim Otten led, was the most likely taxa producing toxin. So it's common and it is likely producing anatoxin in both the Scott and the broader Klamath Basin. Oh shoot, the dots went away on this. There's a lot of points behind these two lines here. And you can see the R² values, so of 0.8 and 0.7. So they're nice tight relationships where those dots are not existing. So ANA-C gene copies predicted anatoxin concentrations quite well. And this we saw both in the Klamath study and the Scott River study. ANA-C gene copies, which is that gene necessary for toxin production, is on the x-axis, and the actual toxin concentration is on the y-axis. And we have these really good correlations between these two metrics. These gene copy assessments in the lab, they're a quantitative PCR measurement. They cost about half as much as the toxin measurement. So just for being able to sample more affordably to expand your sites, so far we found that it's a great method and can indicate if anatoxins are there and help expand sampling coverage. We tested these drift nets, which we really wanted to try to catch chunks of floating algae moving downstream. That was the point of using them. These are invertebrate drift nets, but if anyone's used them to capture invertebrates in the Klamath, you know they catch floating algae very well too. And so we set these up both in the Klamath study and at some sites in the Scott River study. And we also found that the stuff we were catching in these nets generally represented what we were collecting from algae mats on the bottom of the river. And so this is just another tool that can be used to study anatoxin. If you're not able to actually get out and wade around in the river, if you can't see the bottom of the river, or if you don't know what this stuff looks like and aren't sure what to pinch a little pinch of and put into a bottle. We did find in the Scott Valley that it was not very useful for small streams because it caught a lot of leaves and sticks and other terrestrial material. And so it can be useful in some situations, but selectively. So it does take a little bit more time and effort than just going to the stream, grabbing a chunk of algae and sending it in. Okay, another, oh no. I'm getting it too, Kate. So there's a lot of dots on these as well and some lines that disappeared. But the take home points are here either way. So spat samplers are these really cool little sachets of resin that absorb the toxin. So you can put these out in the river and they just sit there passively for about a week is what's recommended. And they absorb the anatoxin, and then you can send them in for toxin analysis. So we had spat samplers out, and these graphs are showing either the ANA-C gene copies or the toxins on the left and right, and how they compared from the mat material and how they compared to the spats. And you still get these positive relationships where the spats that have higher toxins are at sites where you have mats with higher toxins. But so they're very easy to deploy. You don't need any identification skills to use these, and you can still get a measurement of anatoxin in the stream. But we did find that they were a lot less reliable than a person going out to the stream who was willing and interested in getting wet, looking for it, and knows what to look for. And so if you have somebody that's really familiar and willing to take the time, they're probably going to do a better job than these samplers. So again, they're a useful tool in some situations. Our overall detection rate, we had about 30 paired samples, and about 20 of them detected anatoxin when we found it in the mats. And it was like 8 or 9 of them did not have anatoxin when Bonnie found them in the mat material still. So useful. But if you can train somebody and they can take the time to actually look at the bottom of the river, that's going to be the best bet. At least that's what we're finding so far. Okay, so some seasonal trends. We found that anatoxins were highest in late summer and fall in the Scott River watershed. So this is the 2022 data on the left and the 23 data on the right. The 22 data was 6 sampling sites that were sampled every 2 weeks. And so you can just see the distribution of the toxin data there, relatively low early in the season. So early in the season was late July. That's when we started the study. and it increased during the 4th sampling event in September and stayed high through September and into October. And then in 2023, we just had 2 sites that were sampled every 2 weeks for a total of 7 sampling events. And those 2 sites had a similar pattern where it was low in July and then increased later in the summer and stayed high through mid-October when the sampling ended in 23. We did sample fewer sites in 23, but we sampled, or sorry, fewer sites every 2 weeks, but we did sample more sites, and I'm going to show that in a minute. In 2022, we found anatoxins, well, in both years, we found anatoxins were more common, or at least higher in the Scott River than in the tributary sites. So this is an interesting kind of different finding than the Klamath, where we found more anatoxins in the higher water quality streams. Here in the Scott River, our higher water quality streams are now these tributaries, and the Scott is sort of the warmer, more impaired stream. And so here what we found was that these sites, these stream kind of tributary sites, which were Shackelford, Kidder Creek, east fork of the Scott River, tended to have lower toxins than the Indian Scotty, Jones Beach, and Red Bridge, so main stem sites. And we found similar findings in the 23 data, where all of these sites in the green box are the smaller tributaries. And with the exception of Kidder Creek, which actually had moderately high toxins in it, the Scott River main stem sites tended to have more anatoxin than the tributaries. And so in general, what we found in the Scott River from the Quartz Valley Indian Reservation sampling effort and study generally confirmed what we found in the Klamath study, with a few exceptions that I have bolded in here and underlined. So we think these drift nets are useful, but most useful in large rivers. So I'm going to add that to some of our findings that can inform monitoring. In terms of the types of streams where we're finding these anatoxins, in the Klamath, we said that there were actually pretty high anatoxins in these higher water quality streams. And I'm going to revise that to say kind of in these moderate water quality streams, at least in terms of the Klamath Basin, because once we get into these tributaries that are less impaired, we are finding quite a bit less of it. And then I'm going to add a 6th point that spats can be useful, especially when field ID is not possible, or when you don't have the time or ability to really do like an area search for the actual benthic mats. So where do we go from here? We know that the cyanobacteria are quite widespread throughout the Klamath Basin and throughout the Scott Basin. They're probably quite a bit more common than previously expected. And so now that we know that they're in many of these streams, we need to start to better understand what's promoting them, so that we can start predicting where we're going to find it, where we should be sampling, or where we should be presenting more information to river users. Quartz Valley Indian Reservation is planning to continue sampling in 2024. Also, Dre, who's here from UN Reno, is going to start a study to look at more of these factors that are really promoting anatoxin. She's working with Quartz Valley Indian Reservation to try to start including some of the drivers, and to see if she can find relationships in these streams with the different things that we think might be causing anatoxin production. And again, knowing that it's everywhere, knowing that it's in many places, it's a good time to just start being more communicative about it, taking advantage of the signs that the water board presents, and just making sure that if folks are at the river with kids and dogs, that they know that it is something that we need to be aware of and need to watch, especially small children and dogs, and make sure algae isn't being consumed, because it's really those algal mats that have those thick toxins in them, and that's where the risk is coming from in these rivers and streams. And with that, if there is any time, I'm happy to take some questions.
Yeah, I think. Yeah.
I'm going to start on the left, maybe just for the microphone.
Eli. This is awesome. Great presentation. So I've been in the Scott River doing other sampling 2018, 2019, 2020, all the way through to this year also. I noticed 2020 was an especially bad year. I can't identify algae. And it was interesting to see that 2023 wasn't much better, if at all, than 2022. And there's definitely significant differences in flow volume. I've been trying to think about like, what's the relationship between winter flushing flows and summertime? Is there any effort to like get to some sort of understanding of like a good water year in the winter where we see multiple spikes that could be flushing out sediments and algae? There may be a better year in the summer, or is there any relationship that you've been able to identify between those two things in any place?
I mean, I think that's it's a strong hypothesis. And I think, I mean, both from the scientific world and I talk to a lot of people who just know this intuitively too, right? The river needs a big flow to like clean out and scrub all that stuff that's built up. And so I think, yes, there are strong expectations for that. It's hard to see in the data, it's all on the log scale. It did seem like it was a little lower in 23 after that better winter flow. The really hard thing here is we only have a couple years of data. And when you get at that question of how does a winter flow affect algae the next summer, that's an N of 1 every year. So we need like decades of data to really start seeing these trends, just like what Kate just presented in that other data on the Scott River. And so that's where this monitoring that's just getting started is important, but it's going to need to continue for a long time to start seeing these like differences in annual controls on the algae.
Great. One thing real quick, I know we have, been used to dealing with the pelagic blooms in the Klamath River itself from the Pacific Core reservoirs that are gone now. But, what's your opinion on kind of where we're going to be going with benthic blooms in the Klamath now that we have more of a free-flowing Klamath?
Yeah, I mean, there's a lot of uncertainties there. We didn't find super high levels of anatoxin in the main stem of the Klamath River, and they're finding similar findings in New Zealand, where this has been studied most deeply, that these really clear rivers that are rivers that people like to swim in. They're generally pretty high water quality, and cleaner rivers are the ones that have had some of the highest levels of toxins. And so I don't know that the Klamath is going to have a major problem with it. It's also really deep in a lot of places. So except for the margins, it's uncertain what direction it's going to go. I have seen some of it on some of the sediment from the fire. And so there's a chance that some of these fine sediments might help provide some nutrients and let them, increase for a little while. But I don't know that the main stem of the Klamath is going to be the biggest concern for this.
That's good news. Yeah. All right. Other questions?
Yeah, I had a question about stream gradient. If there's a correlation to these lower stream gradient sections where there's less scour potential, potentially being hotspots for these algal mats.
Yeah, I think there are times that we see it pretty thick in these like slow, low gradient streams because it's pretty delicate. So it can, different types are more delicate than others, but it can build up sometimes. But actually some of the findings, again, from New Zealand and just some of the observations we're making in the field, it often does really well in really fast moving riffles too. Like the samples at Jones, this is from Indian Scotty, actually, a picture that Bonnie took that was from like this big, there's some work that's been done on trying to understand why it's doing so well here. But when you think about nutrients, water's moving really fast. So nutrients are coming at it all the time. And so sometimes the fast water actually can promote more of it too, as long as it can still hang on. So again, this is kind of like against a lot of what we know and think about in terms of water quality. And I think one thing when I say that's important to say, like, this is a natural species. This is like something that has been on this planet for a very long time. And we don't know how much climate change or global changes or water use is affecting its distribution, because we haven't been looking long enough to know much about it at all. And so it's really important at this point to just, recognize the risk and to, don't eat the algae. That's sort of the take home message with this right now. Because yeah, it is often in places that, we don't necessarily expect, like fast moving water.
I was curious if you're aware of any efforts related to understanding this in relationship to spawning salmon. being that it's a benthic and producing anatoxin, I'm just curious if there's a relationship there, if anyone's researched that or looking
Yeah,
into
the
it.
information about how this affects, I mean, there's documentations of wildlife, pets, and livestock, and some documentations of it sickening humans, but there's very, very little information about anatoxin effects on other wildlife. And we were just emailing with the water board, and some folks there were looking up articles, and they found like, I think 2 references to fish, and they were like kind of conflicting information. And so I think there's just a huge amount of work to be done yet, but I do know it's a problem. Again, Bonnie in 2022 told me she went out to do spawning surveys, and the fish were like moving around these giant plumes of it because they were clearing the gravel that had been completely covered with it. And so it was just like blowing up in the water. And so that concern is very much there from folks that are tuned in and observing it, but the toxic effects are like almost entirely unknown on fish in general, not only on salmon.
Yeah, yes, Laurel, you mentioned finding evidence of it being present in the Scott, the salmon, and the Trinity. Did you sample the Shasta also, or did you decide not to sample the Shasta?
No, I didn't sample the Shasta. I was mostly working at sites on that first 2021 study that we had access through like established tribal water quality monitoring sites. And so I had gone toward those sites, and the Shasta just hadn't been on my radar at that point. We were really focused on the main stem Klamath, and then kind of added some of those tributaries later. But I'd be super happy to go walk around the Shasta River and look for it with you at some point if you were interested in doing a little field trip. Yeah, and to my understanding, and I think Jake could speak on this more than I could, I don't think that it's normal to test for anatoxin in municipal water supplies unless it's like considered a problem or something that's come up previously. But I don't know, Jake, if you know.
I'm not sure what the requirements are in California. In Oregon, you do test for it if you have visuals of a potentially toxigenic species. So it just, and I don't know what drinking water regulations are for California, but we could easily find out.
But a lot of people might not know what it looks like. So there might be just a lack of, with that we're all looking for planktonic algae.
Right.
Not really aware yet that we should also be looking for these.
Exactly. The focus has to benthic cyanobacteria yet.
Great. And there were a couple questions online. One is about, is eDNA a viable monitoring tool to check for presence? And is there any amount bad or that's tolerable thresholds? Yeah.
So, I mean, essentially this qPCR technique, it's kind of like, eDNA is like looking for everything, right? But this gene copies technique, this cheaper lab technique is essentially like eDNA, but it's targeted. So you could take a cup of water. That's what Tim did in his 2016 study. And then he ran it specifically for that anatoxin production gene, not for like all the genes. But so yes, I guess generally. And then, yeah, the thresholds for California, they're all based on water column. So this is all based on like decades of research that has been very lake focused, not river focused. And so in California, there's a warning, I think they call it a warning threshold, and then a danger threshold, and it's set at 20 and 90 micrograms per liter. But that again is based on the idea that it's planktonic algae, that you're going to go sample a cup of water, and that's what that threshold has been designed to have. So we have to do a lot of kind of back of the envelope math to try to understand how a benthic mat translates into a planktonic cup of water type threshold. So they don't officially apply to benthic mats. But yeah, we could do some math and kind of figure it out if we needed to.
Right. One more question that I'll ask, and then we'll go to Mike Thomas on the web. Is the fall surge in anatoxin associated with cell lysis, or is it something else?
I was just getting some information from Dre, which I could default to you to talk about this. No, but that's just general information. Dre in the back here was just telling me yesterday that she was coming across some research suggesting that when these algal cells get stressed out, might be when they're starting to release more toxins. And there's other research showing that essentially under like higher nutrient conditions, a lot of them maybe grow, but they might not be as toxic. And then when you start to get more stressful conditions, lower nutrients or other stressful factors might be when they start releasing the toxins. But a lot of that is just, I mean, it's not well established yet, but there is evidence suggesting that stressful conditions might cause toxin production to be higher.
Great. And Mike, you can unmute yourself to ask your question.
Hey, yeah, I just wanted to say great presentation, Laurel. This is a great compliment and kind of goes along with other work that we have done in the North Coast region where we're seeing Microcystin, Anabina, and Oscillatoria creating these benthic habs and producing a lot of anatoxins. Yeah, we have developed a tiered approach at the North Coast Water Board using those spats that you had mentioned. And like you had mentioned too, you know, it might be a helpful tool deploying these spats to look for increasing biomass and toxicity, because what we're finding is a lot of local partners just don't have the time to get out in the river to look for these benthic cyanobacteria. But yeah, the North Coast Water Board is definitely interested in the Klamath transitioning to a more kind of benthic hab dominated system. So we are interested in working with KBMP partners to potentially implement that tiered approach, like I mentioned. But again, yeah, great presentation, and thank you for sharing.
Awesome. Yeah. And the spats are super great, especially for somebody that's like, you know, just going to go up to the water and doesn't either have the time or capacity to do a full search. I was really impressed. Bonnie found it every time she went out there. The technicians that went out with her, they had the time and focus, and it was a big part of their job when they were out there that summer. So it seems like it's more of a resource trade-off than a better or worse. It's one of the tools in the toolbox, depending on what resources you have to monitor and sample. So yeah. Thanks, Mike, for that.
Yeah, thank you.
All right. Laurel, thank you so much.
Yeah.
You've been a great repeat presenter here at KBMP. So once again, thank you.